Hello and welcome back to Enzymes. This is actually the end of the unit. It's only like 2 lessons. So yeah, it's going to move quite quickly. We're going to talk about what speeds up and slows down reactions and what a feedback cycle is. So that's the plan for today. So you should have these notes out, enzyme function. And for some reason, let me just move this. And here we go. As you can see here, there's a lot of different kinds of enzymes. So often its name is related to what it breaks down. So lactase breaks down lactose, lipase breaks down lipids, protease breaks down proteins, etc. So, but in some cases, the name doesn't make sense. So the learning target is I can predict the effect of 6 different factors on enzyme reactions, and I can explain the difference between lock and key and induced fit model. So just to review from the last class, A is the substrate for enzyme 1 to create B. B is a substrate that works with enzyme 2 to create the product C. Then C becomes the substrate to react with alongside enzyme 3 to create the product D. Throughout the metabolic pathways, enzymes combine with their substrate to form a complex. What does that mean? So here we see enzyme plus substrate, and they're combining together to make an ES complex. What that looks like is here's an enzyme, and they actually physically fit together with their substrate. So they do connect with one another. This is so, okay, remember how like the sequence of amino acids makes a specific protein with a specific shape for a specific function? This is why it matters, because of that. When it has that shape, it can be the enzyme it needs to be for that substrate. So lipase only works on lipids. It will not work on other things. It needs a specific shape. So the enzyme substrate complex is when they are joined together. That is the complex. In most instances, only one part of the enzyme actually joins with the substrate, and that is called the active site. So that's right here. It's like Pac-Man's mouth, if that's what you're thinking it looks like. This area here is called the active site, and that's where they fit together perfectly. They fit together like a lock and key. That means if you take your key, it only goes in a specific lock. You can't take your house key and open the school with it because they do not fit together. They are the wrong enzyme and substrate, okay? The enzyme will have a specific shape to fit a specific substrate. It is now understood that the active site can sort of hug the substrate to fit better, and this is called the induced fit model. So the two models are lock and key, that it's set and only one key can open the door, or there's also the model called induced fit, where the active site is known to be a little flexible to kind of embrace the substrate a little. So it's more now an induced fit model, but many people know it as a lock and key. So lock and key was what we understood it as for the longest time, and now they've said it's more of an induced fit. So it's like saying the lock in your door can kind of like hug the key a little bit. Okay, so here is the complex. So here's the substrate. Here's the enzyme. That's the active site, and it joins with the substrate. They join together to make a complex, and then it actually causes a product. So it will break apart the substrate or join it together. Depends what reaction is occurring. And you can see it's created 2 products here. Whereas the opposite is it takes 2 substrates, they fit into the active site, and then they join together to create a product. And quiz question for you. When you have 2 joining together, what's actually going to come off? I'll give you a clue. It's a form of dehydration synthesis. Did you say water? Because that's it. So this is how it's all coming together from before. The change of shape, the change in shape of the active site forms the enzyme substrate complex and helps the reaction to occur. Once the reaction is complete, the products are released and the active site goes back to its original shape. The enzyme is not used up, so it will work again and again and again, right? When you use a key, you can still keep using the key on other doors of the same house. You don't need to get a new key. So it can be reused over and over again. So enzyme plus substrate makes an enzyme substrate complex. Then the enzyme is unchanged and the product has been produced. Now the enzyme is going to go find another substrate that fits with it. Some enzymes will do more than just bond to their substrate. For example, For example, trypsin is an enzyme that breaks down proteins by breaking apart peptide bonds and bringing in water. Remember, water is going to break things apart. So it's very important that specific enzymes are present in order for reactions to occur. So what enzymes are doing, they're either breaking things apart or putting them together. Basically, they're assisting in the process of dehydration synthesis and hydrolysis. It's not going to happen fast enough on its own. It needs the help of these enzymes. Here you can see a picture of the active site, and the substrate's going to fit in there, but you can see the substrate's pretty small. This enzyme is going to hug it. I hope you don't write hug it on the test. What I want you to say is it's an induced fit, that the enzyme will wrap itself around the substrate. I hope that makes sense. So how to recognize if something's an enzyme? Enzymes are often named for their substrate, so lipid, lipase, maltose, maltase, ribonucleic acid, ribonuclease, so it breaks down RNA, lactose, lactase. This is not always the case, but that's an easy way to remember it. Now, there are many things that can affect how fast a reaction occurs. How much substrate you have to work on. Like, do you have a lot of protein to break down? If you only have one, you'll break it down and then the reaction stops. So how much reactant do you have? Temperature. What do we know about proteins when temperature gets really high? Did you say denature? Because that would be it. pH, enzyme activation, enzyme inhibition, enzyme cofactors. So we're going to go through all of these. So you can see here the rate of the reaction is dependent on a lot of things like temperature, pH, pressure, etc. So let's go through each one. Number one, substrate concentration. That means how much reactant do you have in that cell. So generally, enzyme activity increases as you increase the substrate concentration. As more substrates fill the active sites, more products are produced. And a maximum rate will be reached when all active sites are filled almost all the time. That's like saying you're all enzymes, and the test needs to be written, and that's the substrate. If I give you all a test, and there's 20 of you in the room, I can do 20 at a time. But I still have more tests. So as soon as you're done, I'm going to give you more, 40, 60. The rate of the reaction will be at its maximum rate. So all of you writing. If I get one person to write a test, 2, 3, 4, up to 20, and then you can only do 20, you can only do 20 tests at a time, so that's why it levels out. Enzymes can only work so quickly. You can only work so quickly to write all the tests. So that is what that is saying, is like the rate is going to increase. Like as you add more reactants, it's going to increase, but then it's going to level out because enzymes can't work faster than their fastest speed. Okay, so as you increase the substrate, even if I have 1,000 tests to be written, you can only write it so fast. So that is why it levels off. The second one is temperature. As temperature increases, enzymes will increase their activity because more molecules are bouncing off of each other. You should remember that from like diffusion in grade 8. But if the temperature goes beyond a certain point, they'll start to get denatured, change their shape, and be completely useless. So usually our optimal temperature is 37 degrees. Once you get past 40, you can see it really declines, and then it will stop completely at 65 for sure, because they're all denatured. pH. pH is very similar to temperature. Each enzyme has a preferred pH level where its reaction rate is the highest. If you change the pH, it can change the interactions and can also lead to denaturation. Remember, enzymes are proteins and can be denatured in extreme temperatures and pH. So one thing I wanted to point out here is every enzyme is different. The one in our mouth will function best at 7, in our stomach at a pH of 2, in our intestines a pH of 8. So it really depends where it is in the body. Whereas temperature, it's the same everywhere. It increases to 37 in our body and then it will start to decrease. But pH is dependent on where it is in your body. Enzyme activation. Not all enzymes in cells are needed all the time. So genes can turn on to increase the concentration or turned off to decrease an enzyme. Some enzymes can be found in an inactive form. For example, in our stomachs, we have an enzyme called pepsinogen. It can't do anything, but if we activate it into pepsin, it will break down proteins. So all that's saying is sometimes we have like enzymes stored in an inactive form. It's not ready to go and we need to activate it. Okay. Enzyme inhibition. Inhibition means stop. So enzyme inhibition occurs when the substrate is unable to bind to the active site of an enzyme. What would prevent it from binding to the active site? What happens is, if the shape is changed, it would not bind. Or if something else is filling the active site, it would not bind. So again, I want to read what this says. Enzyme inhibition occurs when the substrate is unable to bind to the active site. So two things can cause this. The shape of the active site is altered, or the active site is full with another molecule. The reason you might want to do this is you might not want everything to go to the final product. You might need to take a break, and so your body will cause this inhibition, so it stops and slows down, so you don't go and waste all of your substrate. Feedback inhibition. If there's a lot of product, so you've made a lot of glucose and you don't need to make any more, what happens is that it can bind to the enzyme active site to prevent the substrate from binding. So here we have an inhibitor. It's filled the active site so the substrate cannot get in there. This helps the product remain in a certain range. This is also known as a competitive inhibitor because it's competing for the active site. So again, this happens when you have too much product, you need to slow down, an inhibitor will block the active site so it doesn't continue with reactions. This isn't the only time it can happen. Most pathways in our cells are regulated by more complex types of feedback, so it might not fill the active site. What happens is it could bond to a different site on the enzyme, but because it's bonding to the protein, it also changes the shape of the active site, and therefore it also will not bond. So if it fills the active site, that is called competitive because it's competing, this is the active site, it's competing for it, so nobody else can get in. If it is joining at a different area and still changing the shape of the active site, that's called non-competitive inhibition. It's not competing for the site, but it's still inhibiting it. Some examples. So cyanide poisoning is like if you've ever heard of that, what the poison does is it stops cellular respiration from occurring. So it stops the enzymes that allow allow cellular respiration to occur. That means we can't use the oxygen and glucose to make ATP. We stop making ATP and we die. So cyanide is an example of something that stops enzymes from functioning. Another example is penicillin. It stops the enzyme that makes the cell walls of the bacteria. So without a cell wall, the bacteria will die. So that's how penicillin works, is it stops the enzyme from making cell walls. So bacteria die. Here's an example of different ways. Actually, you know what? We're not going to worry about this right now. But if you add, you can see that if you add inhibitors, it will slow down the rate of the reaction. That's all you need to know. Cofactor. The co word means with. So many enzymes need an inorganic ion or organic non-protein helper to function. What does that mean? It means that sometimes enzymes need like a vitamin or mineral to help them fit well. Inorganic helpers are cofactors, and organic helpers are coenzymes. Remember, organic means it contains carbon. These molecules assist the enzymes and may even accept or contribute atoms to the reaction. So an example is vitamins. They are coenzymes. They are helpers that are not proteins. And so if you are lacking a certain vitamin, this will actually slow down the enzyme reactions in your body. So that's another reason why vitamins are so important. So here, for example, is a protein that is going to be an enzyme, but it's actually not active right now. But here is a vitamin. It is going to fit in here and allow a perfect fit for the substrate. So a protein and a vitamin can work together in order to make the perfect active site for a substrate. That seems really complex, so I'll say it again. Sometimes the enzyme cannot function on its own. It needs a helper, a coenzyme. And that coenzyme is likely something like a vitamin. It will help form the active site so that it fits together perfectly with a substrate. This is one reason it's important to take your vitamins. So what is in control of all of your metabolism? It's your thyroid gland. So your thyroid gland is located right here, kind of below your Adam's apple, and it looks like a butterfly. It produces T3 and T4. with iodine in your body. If you have high T3 and high T4, you have high metabolism. The thyroid gland also produces a hormone called calcitonin, which helps to control the calcium levels in your blood. So you might not think your thyroid's super exciting, but it really is because it controls all the metabolism in your cells. If you, well, let's see, here's the next part. So in your body, you have a pituitary gland. It's in your brain. It will cause you to release T3 and T4 from your thyroid. So your brain message goes to your thyroid, and your thyroid releases T3 and T4. When you release T3 and T4 into your blood, you have normal levels. And then let's say that you start to use up your T3 and T4, that message is going to get sent to your brain, and it's going to say, whoa, the levels of T3 and T4 are really low. And so your pituitary gland is going to release more TSH. TSH is thyroid stimulating hormone. And that is going to stimulate your thyroid to release T3, T4, and then levels are going to go down. So your brain's going to cause the release of more TSH and more T3 and T4, and then it's going to go down. So this is an example of a feedback cycle. If you're ever feeling really off, like always tired, gaining weight, it could be your thyroid. So they will check for your TSH and your T4, usually not your T3. So I experienced hyperthyroidism. So I had really high levels of T3 and T4. And so it was, my metabolism was nonstop. This was from an autoimmune disease I had. So I lost like 20 pounds in 2 weeks. It was really like, it was a very crazy time because I couldn't sleep and I couldn't stop moving and couldn't stop sweating because my metabolism was out of control. So this looks really confusing, but I want you to understand that it's a feedback cycle. So your pituitary gland releases TSH. So your brain sends TSH to your thyroid. Your thyroid releases T3 and T4, which will go through your body and allow for metabolism to happen. And when the level gets too low, your brain says, I need to release more TSH. Causes your thyroid to release T4 and T3. Now, again, this can be off many times. So you will notice things about yourself. So for example, if your thyroid stimulating hormone is high and your T4 and T3 are normal, you may have some hypothyroidism if it's trying to go high. Like I said, I had hyperthyroidism. So my TSH was very low. It was like not even stimulating my thyroid at all. And yet I was still producing really high levels of T4. The main thing I want you to know between hypo is low. That is when your metabolism is so low. So notice here your T4 and T3 are really low. And even though you're trying, your brain is like releasing TSH, come on, release more T3, T4. Your thyroid isn't listening. It's not releasing the T4 and T3. So this is when your metabolism is very slow and your brain tries to compensate by releasing thyroid stimulating hormone, but your body doesn't respond. So this, again, you would feel maybe depressed and cold and tired and gain weight, brittle nails. Those are some examples. The opposite is when your brain is saying, don't release any TSH, don't stimulate the thyroid, but the thyroid is still being stimulated and releasing these hormones. Generally, people like this will feel jittery, hard time sleeping, really sweaty, losing a lot of weight. One thing to note, if you don't understand any of this, one thing to note is this happened to me, the hyperthyroidism, after I had a child because of my hormone levels. And so just be aware of that after you have a child. If you experience like confusing weight loss, even though you're eating as much as you can, then it could be that your thyroid is off. I know all of this was really confusing. What I do want to say from it though is just, if you look back here, your brain releases thyroid stimulating hormone, which causes your thyroid to release T4 and T3, which will control your metabolism. Okay, that's what I want to get. And if it is overactive, you are hyperthyroid if there's too much T4 and T3. And if it is too low, you are hypothyroid. Okay. And here's some symptoms of it. So for example, as I indicated, like a hypo, so your thyroid isn't working very well, you will probably gain weight, feel cold, run down, drowsy, sleepy, depressed. Your pulse rate will be really low, brittle nails. So I said most of these. And then the opposite would be true if it's too hyperactive. So you will lose weight, but you'll sweat a lot. You'll feel nervous. You won't sleep. Your hands will be tremoring. Oh, my heart rate was above 100 for like 2 weeks. High blood pressure, etc. So they're very extreme, but it's really important that you're aware of these things in your body. So now I'd like you to watch the enzyme video on Amoeba Sisters because it's very helpful. Oh, look, Valentine's card. You are the substrate to my enzyme and nothing could ever denature us. So maybe good for Valentine's Day. So that is it. And I know that last part was really overwhelming and there was so much information in there, but just know that temperature affects it because it will denature. pH is specific depending on where it is. And also the amount of substrate. It will go up, but then it will level off because enzymes can only function so much. There's also inhibitors that can inhibit the actual site. So if you fill it up, nobody gets in there. It can't work. So you can have a competitive inhibitor or you can have a non-competitive where it affects a different part of the protein but still changes the shape so nothing can get in. That was a lot. I know. We'll review. You can do it.